Okay, thanks, Meg. Yeah, so thanks. I think I know pretty much all of you for the most part, and/or a lot of you have heard about our Edgefield project over the years. But the first part of this presentation, we'll just do a really quick overview of the program and the setup, and we'll try to save the bulk of the time for the data that we've collected and giving you some of those results back in a really helpful manner. So I am Nichole Embertson, for anyone who doesn't know me, the Science and Planning Coordinator, and then Scarlett, who's our research specialist at the district. So we'll be kind of tag teaming. So we want to go to the next slide, Scarlett. Let's see, I think there's been a little delay on things. Perfect. So just a really brief overview of what our Edgefield research program is. So this is funded by NRCS, and we work with individual landowners on using their farms as our research facilities. And we're pretty much looking all at overland flow. We've got one site where we're also looking at tile drainage. So that overland flow at sites with comparative treatments on them. The first few years of any site is background data. So we've got all of like handful 2 to 4 years, depending on the site, of data from all of these different locations that we've got. The key element of that data is that there's no bad data. I know it's easy for us to jump to conclusions when we see like, oh my God, look at that high fecal coliform, this is terrible. But we don't have any data. What we have is data that may have high numbers that is showing us areas that need work or things that are not working, places that we can modify, work with that landowner to improve. So it's really great when we get bad data actually, so that we know what doesn't work, which is just as important as what does. And we really are focusing with the landowners on those results to modify our practices or whatever's going on to improve that landscape and improve water quality. We have a data sharing agreement with the landowners, as well as the program is federally protected, meaning that we have a big confidentiality agreement with this. So we will share the data with you. We're sharing it by soil type and by flow patterns, all the things that matter. Please don't ask who the landowners are, where the sites are. Usually that doesn't even matter when we're presenting the data. It's more about the results. And we always try to share this data in a productive way with you, our partners. So that's part of why we collect all this data, so we can provide those results to you. That helps all of us better understand runoff and water quality and parameters from these fields. So next slide. So just to give you an idea of why we created these, so the primary thing that we're looking for at all of our different edge of field sites right now is we're testing seasonal manure application setbacks. This is a big issue. WSDA has been working with us on this for a long time, for getting producers to use the seasonal setback data that we put together based on, best known science from books. And so we've got this guidance that fluctuates through the year, and it's being largely compared to the CAFO setback of 100 feet. And so those are the treatments that we're looking at. And that, as I mentioned, we have a couple years of baseline data at any site before these are installed. So next slide. So this is just to give you an overview of what one of these sites may look like, just so it's in your head. So all of our sites have a paired field where all parameters are the same soil type, manure applications are not, the field crop type, et cetera, et cetera. So they're usually right next to each other. One is a treatment field, one's a control field. What this means is that we get like double the data per site, right? So we've got 2 setups per site that we're able to get that runoff information and data. So it gives us a really big and robust data set, which is also really helpful. But just to give you an idea of what that looks like. Next slide. Some of the secondary objectives of that. So while our primary objective is looking at nutrient management and in particular setbacks along with other management, these are more generic, and this is the stuff that we want to present to you today, not necessarily the results. And we'll have just one slide to show you how that's going, but we want to share what are specific risk factors for runoff, soil type, rainfall, topography, the big stuff that we can then extrapolate to the larger area of Whatcom County to help us better understand that water quality data, and or this collecting data for local watershed scale modeling, so improving that so we can understand the science, and coordinating with NRCS and other agencies so that we can improve those practices on the landscape on a larger holistic. We can understand how soil types create runoff and when, so any practice that we're looking at, we can better understand how those things are connected on that landscape. Next. What we're collecting, as I mentioned, obviously, we're looking at the farm practices, but we also have that surface runoff monitoring. That's that water quality parameter. We look at soil moisture, rainfall, forage yields. That's more on that farm economic side of things. We've got one site with drain tile monitoring. In other words, we're trying to collect holistically all the data possible for, what is that rainfall? What is that soil doing? When is it running off? How do all these parameters correlate together with the intrinsic soil properties that are there and the slope of the landscape? And then how do we extrapolate that out to water quality and what that might look like on a larger area? Next slide. And the generic, I guess we didn't have it. So the generic field setup that we've got too, the previous slide was just looking at that runoff, and it all transitions into a flume, and that water is collected. All that water sample that we've got goes to the lab, and then Scarlett will now share the results of what we've got. And as I mentioned, we're going to give you those general results based on the years that we've got that are talking more about when does rain create runoff, when do soil parameters create runoff, when do they not? So things that are going to be applicable and usable to all you guys right away. But before we jump into results, any questions just about that background piece? Knowing I'm really going over it quickly so we can really keep our time for the results section. But any questions so far? Okay, so seeing none, Scarlett, I'll turn it over to you and we'll go over the results part.
Cool. Yeah, thanks, Nichole. So like Nichole has said, we've going to be showing you some general trends today that we think will be interesting to the people in the PIC group, and we welcome question and discussions as I go. You can probably say something to interrupt me because I can't see your faces right now, but yeah, feel free to ask questions either during or at the end. So to start, I'm just going to talk about some seasonal rainfall and runoff trends. And so I've stacked 3 figures on here, on top of each other here for comparison. And so the top figure is just the average monthly rainfall. The second figure in yellow is our average monthly runoff volume in gallons per acre. And then below that is the average individual number of runoff events that we've observed at our sites. And this site, I picked this muck soil type site because it's a site that I have the most data on. We've been doing our monitoring there the longest. It's pretty flat, and muck soil type is poorly draining. And you could say that this is probably like worst case scenario in terms of a soil type that is just water's going to run off of. So just to kind of walk you through some of the trends that we're seeing is that in the fall, the rainy season starts here either in September or October. But as far as runoff season goes, we haven't observed any runoff events that occur in September. Runoff events tend to occur at the end of October, mid-October, and then into November. And what this trend tells us is that these soil types have a pretty large water holding capacity and a large capacity to absorb that rainfall coming in the fall. And it takes it a while for it to become saturated and run off. However, once it starts to run off in late October, early November, it tends to increase by month. We get our most runoff occurring in the winter, and particularly in the month of January. This is when it's darkest, the plants aren't growing, the soil saturated, we might get some snow that sits on the ground, etc. So that's when we see our highest runoff amounts. Moving into the spring, we see the runoff and rainfall amounts both decrease, and usually in April we stop seeing any runoff events occurring at all. And I'll also just note that if you look at March and April in terms of runoff, you see that the volumes tend to get much lower, but that you actually still see a fairly high frequency of runoff events occurring. So the runoff events are still occurring, but they're occurring in just smaller volumes. So next, I'm taking a little deeper dive into here into comparing in between our sites. And so our sites they're all on grass fields that have been historically in grass and corn rotations, but they do have different soil types. And so their similar past history kind of allows us to make some of these interesting comparisons. And on this slide, I'm showing you kind of our 3 different soil types, which would be muck, and then 2 different kinds of silt loams, one that is moderately well-drained and one that is well-drained. And I guess just to start, like, what do we see that's similar in between these sites? And I think, first of all, we see that the runoff starts to occur in about the same time, October into November. They both peak in January, and, the runoff event still decreases February through April, and then it ends pretty much in April. However, we do see some differences, and this is something that is expected to see. And so in particular, the silt loam soil type that is better drained takes a while longer to start runoff than the muck soil type. And yeah, you can see that in October, the blue muck soil type is running off, whereas silt loam, shown in orange and green, is not really starting till November. And then within that silt loam soil type, we also see some differences in the timing and the magnitude of runoff, with the more well-drained silt loam running off less, and also the well-drained silt loam tends to stop running off in February, which is, like 2 months earlier than the other 2 soil types. So yeah, I think the take home here is that the runoff volumes are going to vary by soil type, and this is something that I think that we expected, but has just been kind of good to collect this data and see it here happen on a local level. Another kind of larger trend that we're seeing is that the soil drainage classification is a really important determiner of runoff. And on this plot, I've shown you the percentage of rainfall that becomes surface runoff. And I've ordered it kind of by highest runoff to lowest runoff on the x-axis there. And on the x-axis, you'll see that I've noted what the soil type is, so muck or silt loam. I've noted what the drainage classification is, and this is an NRCS soil drainage classification. And then I've also noted what the NRCS hydrologic group is. And so these are just kind of different innate properties of the soil that we can get from data sets. And yeah, we expected that the hydrologic soil group would be kind of the best determiner of if something should run off or not. But what our local information is telling us is that this drainage class actually does a better job distinguishing in between soil classes. And so this is something that here at the district, we're hoping to take into account whenever we think of site-specific risk ratings for recommendations for what practices use in your field. And also we're using this information in another assessment that we're working on, where we're trying to identify fields that are highest risk. And so trying to use this innate soil property in that other assessment is just kind of something that we wouldn't have known if we wouldn't have had this edge of field research. So yeah, that's just kind of a cool thing that we've been able to pull out of here, as this is a really important factor. That being said, you know, soil types do not always act the same. And in particular, we have sites that have the exact same soil type. They're located, you know, very close to each other. However, one of them we've gotten really no runoff, and the other one we have gotten, yeah, some significant runoff in the order of like 5 to 10% of annual rainfall. And the reason for this difference, we think, is just some site-specific terrain factors in terms of how it slopes and how it's contoured. But I think it's just kind of a cool note that, yeah, that there are these trends, but there are always some exceptions to the rule. All right, so now within a year, I'm really trying to get at how much rainfall do we need to initiate a runoff event? Because I know this is a question of what conditions are required for runoff to occur. And as you all know, this is a multi-variable question, and something that we're working through with this edge of field data. But you know, rainfall amount is definitely a big part of that question of what actually causes these runoff events to occur. So on this plot, I've summarized our observations at one of our sites, and I've just plotted only rainfall events where runoff has occurred, and I've kind of showed you this on a monthly basis. And these are box plots, and so just to remind you that middle bar on the box plot is the median, and the box represents the 25th to the 75th percentiles. And then within each of these box plots, all the little dots are kind of all the different events that go into looking for this central tendency of how much rainfall on average causes runoff. And I think I'm looking at this, and I was hoping there would be kind of more patterns. But I think that, you know, in general, we can see that it requires more rainfall in October and then again in the spring to cause a runoff event than it does in the winter. But not like that much more. It looks like it's on the order of like 0.2 of an inch or something on average. And I think another take home of this is that about 1/2 inch of rain seems like a pretty good threshold for runoff to be occurring in vegetated fields during the month of October through May. And so I think this is kind of generally understood, but that's also what my data is kind of showing here. Again, this is just on one field, and so this is likely worst case scenario, but that's kind of the trend that I'm seeing there. So next, getting at our initial, you know, kind of research question for how this project started is, do we see differences in between the control, the 100-foot setback uniform versus the treatment, the seasonal manure application setback plots? And so this is at just one of our sites, the muck soil type where we have the most data. And I'm comparing the control on the left to the treatment on the right, again with box plots. And I've done some significant difference testing, and the only two parameters that, I guess the only one parameter that's coming out as significantly different is suspended sediment for the control. And it also looks like nitrate might be slightly higher on the treatment in this particular year. But I think by and large, we're not seeing really clear differences in between the control and the treatment, which to us indicates that these two practices, whether you're using a control setback or whether you're using a variable seasonal setback, are similarly protective of water quality. So they're not really seeing any differences at this time. All right, so that was kind of like the bigger trends that I feel like, yeah, I wanted to share with you guys today, that I feel like pretty confident about saying, just looking through our data. And now I kind of want to show you guys some of the weirder, like unexpected observations and results that we've seen that, yeah, are just making us think more here at the district, and also things that we're working on, but I think would be interesting to you guys. So the first is we are looking at some patterns over time in fecal coliform, and I would say by and large fecal coliform is really variable, which you guys all know that too, but I think our edge of field results also just kind of hit that home, that it's variable throughout the year, and it's really hard to understand why I get a high event all of a sudden in the middle of January. But one trend that was weird is that we are actually getting some elevated fecal coliform events, fecal coliform concentrations in the spring, even though there were no recent manure applications. So on this graph, you'll see I've put the manure applications here in like a solid line, and those are happening in June and July, but yet in April I was getting counts on the order of 4,000 coming off fecal coliform coming off this field. And I think we don't really know why this is occurring. There were waterfowl and other birds observed in the area, so that could be a source. Another explanation could be that warmer temperatures are increasing biologic activity, and so some of these native fecal coliform bacteria, are starting to grow. I don't know. I think this is just a weird thing we're seeing that, yeah, we're working on, and just kind of brings up again why fecal coliform is just kind of a confounding indicator at times. And so now I kind of want to show you some of our drain tile data that we're collecting on one of our sites. And in this figure, I've got fecal coliform concentrations, total nitrogen, and suspended sediment over the course of 1 year. And again, you can see where the manure applications were in the summer. And I think the take-homes I'm seeing from this are that the drain tiles do appear to, have fecal coliform concentrations in them, as well as nutrients and suspended sediment. And also that we are seeing, again, some elevated counts in the drain tiles, similar to what's happening in the surface runoff. And it's just kind of another confounding thing that we've observed at this site. And so then another kind of, this is maybe not as weird of thing, but we have seen that there are some elevated nutrient and fecal coliform counts in surface runoff after manure application on low risk fields. And I think that the kind of the weird part of this is that there was a good bit of time in between the application and whenever the next, rainfall and runoff event occurred, on the order of like 10 days, or sometimes you can see in 2020 there was, like a half a month in between whenever there was manure applied and then runoff occurred. And our understanding is that this fecal coliform, should not be living out in these cold temperatures on this field, but yeah, we're still seeing that in the runoff. And so we're taking a really hard look at this and verifying our data, and yeah, making sure that there's not something that's just getting our equipment or what's going on with this. But it happened, 2 times now, or 2 different springs. And so this is just kind of something else that we are, yeah, trying to understand better and looking at in our information. So in summary here, the rainy season correlates really well with the runoff season, which is generally October through May, with peak runoff occurring in January. The dry season, June through September, we're not seeing any runoff coming off these grass fields. We didn't generally see that poorly drained soil types like the muck has the higher runoff potential than the well-drained soil type. So your soil type definitely matters in terms of whether your field will run off and how much it will run off. We see a range of how much annual rainfall becomes runoff, and that ranges between 5 and 25% of the annual rainfall actually just ends up running off the field. We also think that local terrain features are an important determiner of runoff. And then, yeah, the last point on here is just that there's still a lot of things that we don't know and we're still trying to understand. So with that, yeah, thanks for having us today. And yeah, let me know if you have questions for Nichole or myself. I think I wanted this one.
Yeah, any questions? We can go back to any of the data slides if you guys have specific questions. You can just unmute yourself and ask. Or your
I've got a question. There was that slide where you were comparing the control and the treatment, and there was one, I think it was ammonia, where there was only a bar for the treatment.
Correct.
Yeah, and that was because it was always non-detect in the other site, and I believe it was detected, yeah, like maybe one time or something in the treatment. So yeah, we're not really seeing ammonia very often in the runoff. So it's kind of hard to compare those two since we just have all non-detects from the control.
Which is good. And you can even look at the values, Hannah. They're like super small. So in other words, it was pretty much always by and large non-detect. Any other questions?
I had a thought about the high counts in the spring, not during manure applications. I don't know exactly the setup of how you guys have it, but I know ditch clearing has been a question that we've kind of talked about. You guys would know that had been happening, right?
Yeah, this is just overland flow from a field. This is not sampling out of a ditch or a waterway. It's purely water off. Yeah, right. So there's like, we're just out there like, what the heck? So maybe ducks or wildlife that are on the field through the winter running through, but it's a confounding one we're still trying to figure out. Because it's high count at both locations, right? At the exact same time. One had manure and one didn't on those two fields. So it's a really good one.
On those dates, I know you guys were doing E. coli and both E. coli and fecal coliform. Did you see any differences between them? Or kind of the same patterns that you see throughout the year on runoff events?
We just were doing fecal coliform.
Oh, okay. Yeah. That might be interesting for future years in terms of better understanding like the soil, how much of it might be soil regrowth possibly as fecals as opposed to E. coli, which in theory should be like more fresh sources.
Yeah. Or DNA is another one we've considered, seeing if we can add that in. So yeah, it's one of those confounding ones, and it's just these little snippets of oddities that were that come up, and occasionally we can figure it out and some not, but it's great data. As Scarlett and I were talking about the other day, as with most research projects, it's more questions than it answers, which is great. And having your feedback too of what you guys might find interesting from a data analysis perspective on some of this, or obviously we're doing our research project, but as you can see, the data as a whole is really helpful for a better understanding runoff in its characteristics and behavior. So please let us know if you ever have questions or inquiries about the data, that we can look at it in a novel way. It's also helpful.
Did you say that you did see the elevated counts in the drain tiles in the spring as well?
Yeah.
Yeah, so you can go back to those two slides, Scarlett, perhaps. Yeah, Dan, same time.
Yeah. I mean, not knowing a lot, you know, like just based on wild conjecture. I think that idea of it just being like reanimated stuff as the temperatures warm up that's already in the soil, like, that makes some sense to me. But
Yeah.
Yeah.
And there's like, they apply manure once a year on this field. So it's not like, it's a tough one to say, well, it's not manure related. So I think there's other sources besides just manure that are in the soil that are coming up. As we all know, fecal coliform is a humongous profile of things. It's not a singular, it's not just species per se. So yeah, I think you're right in there. And you can see total nitrogen and suspended sediment had different peaks and characteristics. So we always try to follow that profile when we're looking, is it manure, is it a different source? And so looking at all of the different parameters that we collect, phosphorus is another one. We're always looking for the relationship, and fecal is really high at a time when nitrogen wasn't. Nitrogen's been much higher at a different time. So it's just looking at like, what is this fecal source that's suddenly blooming at that time period? It'd be really interesting to keep up the microbiology to look at that.
Yeah, I think the other thing that stands out to me is not, I don't have a question about it, but just the that timing, after applications of how long you're seeing those elevated counts is interesting. Like, I think that's really valuable information of like how long it can last.
Yeah, and that's a question I know we've always had, especially in the winter. We're like, well, we're following all the guidelines and science that we have available to us. We're still getting high counts. So like, what is that waiting time? Or is there something else we could have done differently? And is that winter persistence?
I think I missed it. When did you say they were doing the application? They were doing it once a year?
Look at those lines. It's the lines that are on those, so that helps you. So at the other site, sorry, Scarlett, I think it was referenced to that one. So it was like way after. They didn't even know, they applied like the summer before that, over like almost a year ahead of that.
It's bizarre.
Yeah, it didn't, that was not manure persistence that I could gather. Yeah.
Well, these ones too, there's kind of a big gap between that high before, before the manure application and the highs after. Do you only sample after a rain event, or is there a reason why there's no data kind of like right after those manure applications? Just after rain?
Yeah, so we only sample when there's like water flowing off these fields. And so there's just no runoff events that are occurring in the summer on this field to sample.
Yeah, so Hannah, it's hard, I know it's hard to sometimes get your brain off of like river sampling, right? You're like, you can sample anytime. So obviously we only have data when we have runoff. So it has to run off, and we're always sampling when there's runoff. So if there's no sample, it means there wasn't running off.
Okay, I see. This picture really kind of helps me visualize it. Thanks.
Yeah, sorry, I took this out because I was trying to cut it down, but this is kind of like a monitoring point on a field that we're collecting this overland flow piece before it goes through the woods or goes through a ditch or wherever it goes.
Any other questions from folks? Well, like I said, if something pings on you later, you have a question, an inquiry related to the data, the project, or ideas, just holler, let us know. Like I said, we use the data in one way for our results, but this data is very usable back to this group in many ways. So we're always helpful, or hoping we can work together and, amenable to giving updates as we find new things that are kind of applicable to the larger landscape. Well, thanks everyone. Thank you, Scarlett.